Hi, welcome back to my channel. In this video, I will be guiding you through the topic of cholesterol in a very simplified way. This is going to be just like a one-shot video in which everything is covered about cholesterol and most of the MCQs will be answered through this video and it will be an easier way to remember. So I have just put a few headings which will basically guide us through the lecture. So first of all, for the introduction, you just need to remember 2-3 points. So the first point being that it is an exclusive animal sterol and it is not seen in plants. All right, then coming on to the next point, you need to remember that it is amphipathic. Cholesterol is amphipathic. What is meant by amphipathic is hydrophilic plus hydrophobic. It has both the natures. Now, how many carbon atoms are present in cholesterol? There are 27. Daily synthesis is 1 gram per day. All right. Then organs involved. The foremost is the liver, which is 50%. Then we have intestine, which then we have skin. Important adrenal cortex and reproductive organs. Now, site is the most important question and it is usually asked in the MCQs. So for site, you have to remember two. One is the cytoplasm and the smooth endoplasmic reticulum. All right. Then source of carbon is acetyl CoA. All right. Now the pathway. I will explain the pathway in a very easy form, so it will be very easy and you will just remember it a lot better after watching this. So first of all, let's just get started with the pathway. So first of all, as I told you, the source of carbon is acetyl CoA. All right. So starting from the acetyl CoA, we have what is formed as acetoacetyl CoA. All right. Then we have, first I'll write the substrates. All right. Then we have HMG CoA. Then HMG. Then basically I'll write the enzymes as well. So in acetyl CoA to acetoacetyl CoA, we have what is called as thiophorase. Then from acetoacetyl CoA to HMG CoA, basically we are forming HMG CoA, so we have HMG CoA synthase. Then we have HMG CoA reductase. Now, in HMG CoA reductase, then we will have mevalonate. Now you need to know that mevalonate is 6 carbon. Okay. Then we will have isoprenoid units. They are 5 carbon. All right. Then we will get geranyl, which will be 10 carbon. How we will get it? By isoprenoid unit, again combining. So it's like 5C, 5C, 10C. Then again we will see that isoprenoid unit will again combine and we will get a 15 carbon farnesyl PPI, which is 15 carbon, okay, with inorganic phosphate. Then 15, then we will see that this will combine again with farnesyl PPI 15 carbon and they will form squalene. All right. Then this squalene, basically here what you will see is the enzyme is squalene synthase. Now this squalene synthase, squalene is formed, which is 30 carbon. Okay, 15, 15. Now we have 30 carbon. Then we will get lanosterol, which is 30 carbon again. Now there are many changes that happen from squalene to lanosterol, which I will be telling you further. But now we are just forming the foundation. Now this lanosterol will form what we need is, just give me a second. It will form. So it will form cholesterol. All right, 27 carbon. Here also changes are occurring and here also. I will tell you after some time. But now you just need to know this much. Now, so in this whole pathway, there is only one ion you need to remember, which is magnesium. And you need to remember NADPH, okay, which is needed. then you need to know that this is our rate limiting enzyme and this is our committed step. Okay, now let's just talk about it. Now, how is NADPH coming? Now, you all know that NADPH sources basically are HMP pathway, right? HMP pathway. Then we have the malic enzyme, and we have the cytosolic isocitrate dehydrogenase. Okay, then RLE, I told you it is a very commonly asked question, so you should just like, now it will be basic for you. So HMG CoA reductase. Now I told you to remember one ion, which is magnesium ion. Now the relation of insulin and glucagon to cholesterol synthesis. Now I want you to think if a person is eating well, obviously it is then well fed stage, right? So in well fed stage there will be increase in cholesterol. Now when is a well fed stage seen? Basically when there is insulin increase, right? So well fed stages, insulin increase, so insulin will increase the cholesterol, whereas glucagon, it will decrease. Okay, so increase and decrease. Then any hormone which is related to it, so we know thyroxine. I will tell you how this is happening in further video. Thyroxine basically increases the cholesterol synthesis, whereas glucocorticoids decrease. I will tell you how this occurs. This is basically related to the phosphorylation and dephosphorylation. Now, for now, just know this much. Now, in statins, what are statins? Basically, statins are the reversible competitive inhibitor. Basically, they are the structural analogs of, now you just need to know ki what are they inhibiting. They are inhibiting HMG CoA reductase. All right. Now, regulation of de novo synthesis of cholesterol. Okay, just give me a second. Now, in this, you should know basically 4 points. Now I will tell you and how it is happening. So first of all, basically there are 4 mechanisms through which the regulation of the de novo synthesis of cholesterol occurs. So starting from the sterol dependent. So first is sterol dependent regulation of gene expression. Okay, then we have sterol accelerated enzyme degradation. Okay, then we have sterol independent covalent modification. Then we have hormonal regulation. Okay, just know these 4 points. Now what do I mean by all this? So we need more space. So first of all, I want you to know that this one is long-term regulation. So long-term. Okay, now see, I have something like, okay, so basically I have smooth endoplasmic reticulum. Okay, now the smooth endoplasmic reticulum, it normally has SERP and SCAP. Now what is SERP and SCAP? SERBP and SCAP. Now SERBP is basically sterol regulatory and element binding protein. Okay, and SCAP is sterol, so it is basically SREBP, right? And SCAP is SREBP, cleavage activating protein. Just remember the short form, that much is enough. Now what happens is basically we have smooth endoplasmic reticulum. Now when there is decrease in cholesterol, okay, so what happens is this then we have Golgi apparatus. So what happens is this smooth endoplasmic reticulum transfers the SREBP and SCAP to the Golgi apparatus. So here comes our SREBP and SCAP. Now what happens is here we have basically um, then we have over here nucleus. Okay, now what happens is in Golgi apparatus the SREBP and SCAP is formed, right, is transferred. Now what you will see is here we will have proteolytic cleavage, basically, yeah, proteolytic cleavage. Now due to which we will just get SREBP 2 in nucleus. Now normally you have DNA in nucleus. Now this SREBP will cause mRNA to form, basically transcription. Then this mRNA will further translate and form HMG CoA reductase. Now this HMG CoA reductase, basically it is the rate limiting enzyme, so it is increasing, so synthesis of cholesterol will also increase. Now when body gets normal, what happens is this HMG CoA reductase, it will bind with the SCAP, all right, and it will bind with the SCAP, and SCAP will also bind to some other proteins. So basically when normal achieved, then cholesterol binds with SCAP. SCAP also binds with other proteins, others like INSIG, very important. So this is insulin inhibit induced gene. Okay, so basically when it binds, then what will happen is they will prevent the release of SREBP and SCAP from smooth endoplasmic reticulum. Okay, now let's just once again go through this. Normally SRBP is present in our smooth endoplasmic reticulum. Now when we see that there is decrease in the cholesterol, then we see that it is moved to the Golgi apparatus, and after Golgi apparatus it undergoes a proteolytic degradation. Now just due to this proteolytic degradation, we have ki SREBP and SCAP will now like be cleaved, okay, broken. So SREBP will go into the nucleus. In nucleus it will cause transcription, then translation, and it will increase HMG CoA reductase, which will further increase the cholesterol synthesis. Okay, so this is what is sterol dependent regulation of gene expression. Now in sterol accelerated enzyme degradation, what happens is when there is increase in cholesterol, so HMG CoA reductase binds to INSIG, okay, which causes the ubiquination and proteasomal degradation of HMG CoA reductase, so decrease in cholesterol synthesis. Now talking of these two, sterol independent and hormone, now just know that when we talk about the HMG CoA reductase, now HMG CoA reductase, okay, and HMG CoA reductase, so basically when it is dephosphorylated state, it is present in dephosphorylated state, it is its active state, okay, but HMG CoA reductase in phosphorylated state, obviously opposite, right? So phosphorylated state, it is inactive. Now if you see, this is inactive and this is active. Now you need to know that the phosphorylation, the thyroid, basically thyroxine, now phosphorylation. Now you need to know what causes phosphorylation. Now phosphorylation is caused by glucagon and glucocorticoid, and see this dephosphorylation. Now dephosphorylation is caused by thyroxine and insulin. So this is the reason why these increase cholesterol and these decrease cholesterol. Okay, now you know the answer why we chose these answers in all this. All right, so this is it. Now in specialized products of cholesterol, so I hope regulation is easy. We will cover the next part in the other video. So thanks for watching, and do watch the second part.